Oh, let's call her A. Question is probability of a O child. There's a test question, final exam. Do you remember ABO blood type? Can you figure it? No. Okay, look it up. I know you have it. So, is that phenotype or genotype? I did the same thing 3 months ago to you guys on the board. All right? This is phenotype. O, A, B, and B are phenotype. Genotype are the genes. Little I, little I. We don't know whether mom is homozygous or heterozygous for the A blood type. So how many squares do we have to do? 2. Just work them out. Adds up here on top. We'll do this. We'll do this. We don't know what mom is. When you recombine, probability of an O child, 2 out of 8, 25%. Probability of an A child, 75%. 6 out of 8. Probability of a B child, 0. Probability of an AB child, 0. You have 2 weeks. Go back and do your blood typing again. Figure out your genotypes for A, B, and O blood types. And do some problem sets. Guess what's in your lab manual? There's a whole chapter on heredity. It's in the back of your lab manual. And it has Punnett squares. And it asks you these questions. Huh. What's the probability I throw a heads? The same thing. 50%. I throw a heads. Tails. Was it 50% I throw a tail? What's the chance I throw 3 tails in a row now?
0.5 to the 3rd.
Chance is probability to the nth power. 0.5 times 0.5 times 0.5. I've got about a 5% chance. I'm doing this in my head. Did somebody punch that in for me?
12.5.
12.5. Thank you. I know it didn't sound right, but I wasn't using a calculator. 12.5% chance that I flip 3 tails in a row. Should we go for it? Probability I throw a tails right now? 50. Chance? This is my second event. 0.5 times 0.5, which should be 0.25 or 25%. I got a 25% chance of throwing another tail.
Tails.
What's the probability I throw a tails? Probability, single event, 50%. Chance that I throw a 3rd tails in a row now?
12.5.
12.5. Can I do it? Odds are going down.
Tails.
I'm on a roll.
This happened last time, Stan. Is
This
that
happened
that
last-
quarter?
Same quarter. Same quarter. All right, so now I'm on what? My 4th round? 0.5 times 0.5 times 5 times
6.25.
6.25. Thank you, Cliff. I got a 6% chance I'm going to throw a tails. What's the probability I throw a tails?
50%.
50.
Tails.
Tails. I'm heading to Vegas.
I see the head, make sure it's real.
Smart. question. You heard of the two-headed nickels, all right? It's a head and a tail. So what have I thrown? 4, 5?
Turn 4.
Next up, we have 1.5% now.
No, we're at 3.
3%.
3 point something. 3.125.
Heads, and we're done. We're done. So the odds chance that I threw 4 in a row was down around 6%. Yet in every event was independent of the previous one. All right, that's what you believe. So when I talk about the probabilities and chance up here, you have to use the same odds. Is that if I give you the parents, give me the probabilities and the chances of multiple birthing events. Are we clear on that? Okay. Good. Chromosomal aberrations. Sometimes things don't work out perfectly. In separations of the chromosomes of the gametes, sometimes the pairs don't separate as they normally should. You get 24 or 22 in a sperm or an ovum. You give monosomy XO, as in Turner syndrome, that means 2n equals 45. You give a trisomy 21s or Down syndrome, thank you, Shay. Trisomy 21, it means there's 3 chromosomes at the 21 side. Normally we have 2, that's 2n equals 47. That's called Down syndrome. Klinefelters. XXY is due to a non-disjunction of the gametes. Non-disjunction of the gametes. 2n equals 47. Can they do fetal testing? Yeah, Eli?
What does that look like?
What? Klinefelters?
Yeah.
Used to show these pictures, found that some people are bothered them. They don't have them up anymore, but you can look up anything you want. The way that they go, good, they took the pictures off of this as well. Sometimes can be a little bit disturbing, but
Images on the bottom.
let's see.
Scroll down, there's images there.
Images, thank you. Oh, these are not really pictures of individuals. Usually their genitalia is disfigured. They are typically non-reproductive. There are phenotypic characteristics with the head, the hands, and the body stature. And that's what it looks like if you're doing a genotype, where you look at down here, normally you have 46 chromosomes, but with Klinefelters you'll have XXY. Turner syndrome. Have different phenotypic characteristics with Turner syndrome. The neck, in this case, what they're trying to show is the neck there. It also gives you an idea of one chromosome at the 23 sex chromosome. You only have 1. You're supposed to have 2, and you can get many different phenotypic results of this disorder plus heart disorders. Usually valve replacements are required if a person with Turner syndrome is going to survive. Fair question. You can look any of these up. Tay-Sachs? Awful. I put Tay-Sachs earlier. You guys heard of Tay-Sachs before? Yeah, Tay-Sachs is usually, can't really say that's too much accurately anymore, but at one point it was considered a Jewish ancestry disorder. Patients usually don't make it past the age of 5. And it's a traumatic death, very painful, short-lived life. Was passed in the Jewish ancestry community generation to generation, but now as there's not much of what you would call a pure genetic race anymore, it's not considered a Jewish disorder. It is just a genetic disorder, and that is Tay-Sachs. That's on your list that you should know about. If you have problems in your family or have had previous genetic conditions in your own family, fetal testing is possible. Amniocentesis can take place at 14 to 16 weeks. Some people consider that to be too late in the pregnancy to do modifications, whether that be genetic or termination. There's a new technique now that's called chorionic villi sampling, and it can be done as early as 8 weeks, where they can actually do remove the fetal cells and do genetic testing on them. Let me show you a picture. Here is a karyotype. We actually get chromosomes, put them on a computer, you're looking at them in a microscope, and you can actually look at them. If you count these up, 1 pair, 2 pair, 3 pair, 4 pair, how many pairs should be up here? 23 pairs, 23 pairs, and each pair should have a matching replicant next to it. This is a male or a female? This is a female because we have the XX there, and this is the 23rd pair. This is called a karyotype. You guys ever seen them? Y'all have them. My cytogenetics class in undergrad, Dr. Pensay, 426 cytogenetics. The final exam, I had to walk in with this on a t-shirt, and it was mine. That was the whole class project. It was a graduate seminar, and we did lab work, and we got up, and I remember back in the day, this was quite a while ago, stood on a desk with a tiny droplet of my blood that I had been working on for 4 weeks in the laboratory. Went to the health center, drew our own blood, got it through, ran it through, dropped it on a slide on the ground, and it caused the chromosomes to splatter. Put them under a microscope, took a picture of it, took it down to a local t-shirt shop, and this was the fun. If you could walk in with your own chromosomes, and he knew, because he was our professor was watching us and helping us do this whole project. He knew if they were ours or not. I had a X and a little fragment down here, because of mine was XY. That's called a karyotype. And this is the initial screen that they do when they're doing genetic testing, fetal testing for new babies now. They do these just standard, a triple marker screen. Anybody heard of that? Triple marker? No? Haven't heard of that? Oh, okay. If a new pregnant mom goes into the physician, they will do a triple marker screen, which is actually testing for Down syndrome and 2 other disorders that can be done on a very simple blood test. And that is a marker that they will use now, pretty standard for any new pregnant mom who's getting health care. Good. Uh-oh. Hormones, let's finish. You guys wanna go? Got like 5 slides on it, and he's like, nah, we're done. We're almost done, let's just hang in there. Hey, look, all we got left is like 4 slides here on hormones, and then you're out of here. All you gotta do, and then Wednesday we'll do genetics, so we'll make sure our worksheets are done, and then we'll review next week. That's good. Hormones, chemical messengers. They're usually steroids or proteins. They travel in the bloodstream, they bind to receptors, they're potent, and they're short half-life. What you need to know about hormones is on the hormone worksheet on Moodle. Right off to the side, see hormone worksheet on Moodle. I have collected all the hormones we covered this year, and I've asked you to know what they are, where they come from, what they target, and what they do with the target. There's a hormone worksheet. You fill that out, it'll be a piece of cake. You need to know that hormones have a negative feedback system. If it's positive, it's cancer cells, uterine contractions, or diabetes. We talk about this in AMP1. They're stimulated to be released by hormonal, humoral, or neural stimulus. These 3 are on the exam. I'll pretty much tell you next Wednesday what endocrine questions are on the exam, but I know these 3 will be. I haven't written it yet, but I always put these on. The 3 stimuli for hormonal release in our body are right there. Mechanism of action. If they're the protein or amine hormones, not the steroids, they actually get to stimulating the inside of a cell by binding on the plasma membrane. You use a second messenger. Here's how that works. I'm a hormone, I come up. Demi, that's yours, all right? Pass it to Clifford. There we go. Did I go inside the cell? No. I'm an amine. I bind to a receptor on the outside. You're my second messenger. You give the message to Clifford, and he's going to do whatever my hormone tells that cell to do. That's called a second messenger system. The other way is steroid hormones, and they go right inside the cell. They can inhibit or stimulate, the second messengers can do whatever they want to do to that cell. Secondly, direct gene activation, steroid hormones, receptors inside the cell, goes just like this. I'm a steroid hormone now. Could somebody name a steroid hormone for me? Who we're studying today? Anything? Progesterone, estrogen, testosterone. I go right inside the cell. And I get Clifford the whole box. That's how steroids hormones work. They travel, everything travels in the bloodstream, but the activation at the cell is a little bit different. There's two different ways, second messenger or direct gene activation. For the final exam, you'll need to know specific hormones, organs they're derived from, complete the endocrine worksheet for relevant hormones, and I'll tell you which questions are on the exam when we do the review next week. Are we good? Draw a line. You just finished A&P. Congratulations.
Thank you, done.
Wednesday, if you want, we're in good shape. Did we miss anything this quarter? I don't think we missed anything this quarter, all right? I didn't get sick. Actually left Sun River this morning at 4:45. Get back for you guys today. Beautiful sunrise. Oh my goodness. Over Odell Lake this morning, I thought of you, went by Willamette Pass. No snow whatsoever. Gorgeous drive back. Wednesday, we're gonna work on the genetics, 5-point genetics worksheet. There's absolutely no excuse not to get that all correct and turned in, because I'm giving you a whole day in class to do it with me here, and you can work as a group. So if you blow off Wednesday and you forget about it and you show up next Wednesday and it's turn-in day and you forgot, that's a bad choice. Show up Wednesday, let's get it done. It's 5 points for A&P. You walk out of here, you got nothing on your mind for the weekend. I review next Wednesday for our exam. We're done. We good? All right, genetics on Wednesday. Good luck. Get out of here.